She is the Doris Johns Cherry Professor of Biology and Medicine at the University of Rochester. She's also co-director of the Rochester Aging Research Center. Her research is focused on the understanding of mechanisms of longevity and the genome stability, and on the studies of exceptionally long-lived mammals, such as the mole rat. Dr. Gorbunova earned her B.Sc. from Saint Petersburg State University in Russia and her Ph.D. from the Weizmann Institute of Science in Israel. Her work has received numerous awards and prizes for a variety of internationally renowned medical research organizations, and we're really lucky at Rochester to have her here. So with that, we're looking forward to your talk. Thanks, everybody, for your patience on this one. And Vera, it's all yours. Thank you very much.
Well, thank you very much for the invitation. Well, I will try to keep it lively. I know it's getting late, and I welcome questions. Well, it's a webinar format, so I actually wish we had it in a regular meeting format, so I could see people. But I will try to imagine your faces as you are listening. Okay, so. The topic will be about longevity and aging, and this is a topic that touches everybody because everybody ages, and this is both a very important scientific problem because we want to understand molecular mechanisms of the process, but it also a very important societal problem, and it touches economics, it touches everything, and this is why I'm so excited to be working in this area. And I will also just briefly mention that at the University of Rochester in 2019, we founded the University of Rochester Aging Institute, and within this institute there is Aging Research Center, and the goal is to both pursue basic science research on aging, and also patient care, and bring together people providing care for older adults together with researchers, and altogether what we want to achieve is longer and healthier life for people in the community and all over the world, because once scientific discoveries are made, it's very difficult, they spread, and we want them to be accessible for everyone. So let me begin my story. I will start with showing this painting, The Fountain of Youth, that is quite a famous one. You can see here old people entering these healing waters of the fountain, and then exiting on the other side, rejuvenated and ready to enjoy life, as you can see. And this has been a dream of people for centuries. People were looking for such fountain. And we, in keeping with this tradition, we're also looking for ways to delay aging process, but using science.
I want to interrupt just a second. Chat doesn't work. Use Q&A to ask questions. Just use Q&A. Thanks. Go ahead.
Hey, so yeah, I just for the joke, Fountain of Youth. People actually, even there are speculations that a part of the motivation for discovery of Americas was that Spanish explorers were looking for the Fountain of Youth. And they actually did discover it. So Fountain Youth is located in Augusta, Florida. There is a Fountain of Youth state park. But of course, I'm not going to guarantee that if you drink from it or bathe in it, it's going to rejuvenate you. But, it just underscores the idea that this has been something people were looking for a very long time. So where else can we find Fountain of Youth? Aside from Florida? Well, we can find it everywhere, actually, because if we look at diversity of mammalian species, we can find animal species with very different rates of aging. From very short-lived animals such as mice or shrews that live 2 or 3 years, we have animals that live much, much longer. For example, naked mole rat is one of the animals that we study, and they can live 40 years or longer. And naked mole rat is the same size as a mouse. It's a small, ugly rodent, but it lives 10 times longer than a mouse. There are many species of bats that are very tiny, but they can live 30 or 40 years. And then, well, we have humans somewhere here because we humans are long-lived. But there is one animal that lives even longer than humans, and this is the bowhead whale. They live over 200 years. So there is this amazing diversity, and we can use it to discover mechanisms of aging and longevity, because if, for example, we can find what's different between a mouse and naked mole rat, then we can perhaps then use it to slow down aging in people. Or if we find what's special about the bowhead whale, we can also come up with strategies to slow down aging. So this has been our scientific approach for many years. Here are some of the species that we studied: whale, naked mole rat, blind mole rat, which is another bizarre rodent that lives more than 20 years. Gray squirrel, we are interested in them as well. So very few people know that gray squirrels can live more than 20 years. And if you think of a rat, which is kind of a similar creature to squirrel, but rats can only live 3 to 4 years. So again, this huge difference that we can use for our exploration. And we also have bats that are very good, not only for their long life, but also in a way they can resist various viruses and infections. So once we find out specific mechanisms, and I will mention some of these because we discovered this one and this one and this one, and some contributed here. So once we discover a mechanism or particular gene that's important, we can put this gene in the mouse and say, okay, does it make the mouse live longer? And if it does, then we can start looking for various pharmacological ways how we can mimic the same situation in human patients, and that already opens the path to the clinic. So this is a general concept, and we were actually quite successful in taking it almost up to here with the naked mole rat. I will talk briefly about that. We are also in the process of studying whales, so I will show you some short vignettes about this approach. So let's begin with the naked mole rat. So here is the naked mole rat on the cover of Nature magazine. Also in that year, 2013, naked mole rat was named Vertebrate of the Year because of the discoveries by our group at the University of Rochester. And one of those discoveries had to do with amazing cancer resistance of the naked mole rat, because not only they live for 40 years, but they also are resistant to various diseases, mostly age-related diseases. They almost never get cancer, they don't get heart disease, they don't get arthritis. And what we found is that the reason behind at least some of those amazing properties is this molecule. This is the chemical formula. It's called hyaluronan or hyaluronic acid. And if you are not familiar with it, you probably seen it as a component of various cosmetic creams, because it's a very hydrophilic molecule. It absorbs a lot of water, so it's a very good moisturizer. But amazingly, we find that naked mole rats are full of this molecule. Hyaluronan is a natural part of our tissues. We also have some in our skin and other organs, but naked mole rats have 10 times more. And this molecule gives very unique properties to the tissues of naked mole rats. So this is what we discovered 10 years ago. Then the next step was to take this gene that's responsible for making hyaluronan, or this long molecule, and we put this gene in the mouse. So we made mice express a gene from naked mole rat. And now the question was, is it going to benefit the mice? First question we asked was, will it help them resist cancer? Because mice get cancer very frequently, even more frequently than humans. So if in human cancer mortality is about 25%, which is still high, we would like it to be zero. But in mice, it can be up to 90%. So mice are very susceptible to cancer. And what we found when we put this gene into mice, they got less cancer. So they didn't become as protected as naked morats, because naked morats almost never get cancer. But they were getting significantly fewer tumors. And then the most exciting part was that these mice also lived longer. So this is what we call a survival curve. So this is percent survival in the beginning of the life of the mice. 100% of them are alive, and then as time goes on, they start dying off. So you see the curve starts going down, and by 160 weeks, all mice are dead. But you can see that those mice that had the gene from naked morats, they were aging more slowly, and they were dying less. So it was not, they didn't live 10 times longer like naked morats, but they lived about 10% longer, which is still quite remarkable, because, well, if we can make people live 10% longer, that means people would live 8 to 10 years more. I mean, that would be a big deal for many people. So that was quite exciting. So now we know if we take this one gene from naked morat, we put it in mice, it helps them. They get less cancer, they live longer. We also found that they get a lot less inflammation. And inflammation is one of the drivers of various age-related conditions. So reducing inflammation was also very important. So here is one story from an ugly rodent. We try to understand why it lives so long. We find the gene, we put it in mice, and we make them live longer. So now how can we move it to people? So here, this mouse was transgenic. So we don't want to make transgenic people. But we can develop medicines, drugs that would do the same thing, because the goal is to make more hyaluronan in tissues. And there are some inhibitors, some small molecules that would inhibit degradation of hyaluronan. And this way we can also make more. And right now we are testing some of these molecules. We have some preliminary data that if we give these molecules to mice and we also give them tumors, so then the tumors spread more slowly. So we can actually just take some small molecules and achieve very similar result. We don't know if the mice would live longer. We haven't done that experiment, but we know that we can slow progression of cancer. So here we almost went the full circle. So now let's switch gears, and I will tell you another story. And this has to do with some of the underlying mechanisms of the aging process, because what drives aging? This is a very big puzzle. There is still a lot of debate. But one of the very important contributing factors into aging process is DNA damage and change in the epigenome, because genome is our DNA. But then DNA, it's a very strong, very long strand of nucleic acid, very long. And it needs to be packaged somehow in the cell. And the way it is packaged, it has to be accessible, because sometimes you need to activate certain genes, you need to keep certain genes silent. And that is also very important for the way cell functions. But DNA gets damaged all the time from various external factors such as UV light, X-rays, also because of just the life of the cell. It needs to be replicated, it needs to be opened and closed, and that damages everything. So we have various systems to repair the damage. One of them is called double-strand break repair. So because if double-strand break is like if you break the strand, and it can lead to loss of big chunks of the DNA molecules. It can also lead to these translocations or rearrangements. That is something we find in old cells. We also find a lot of this in cancer cells. And all of these processes together contribute to so-called epigenetic drift or epigenomic drift. And like drift, something is drifting, but what is happening as we get older, this sequence of the DNA gets mutated, and also the way it is packaged gets disrupted. So another way to look at it would be, this cartoon that I drawn. In the young cell, DNA is arranged very neatly. There are some areas that are more tightly wrapped around. There are some more open areas. But as we get older, it unravels. So if epigenetic landscape in the young cell is sharp, there are some regions that are open, closed. In the old cell, it all smoothens. So instead of certain genes being active and certain genes being repressed in particular tissues, we have it all becomes dysregulated. And that affects the way cells function. And that makes our tissues, less fit. You can also compare it to, these socks. So in the young epigenome, everything is folded very neatly. So when you need certain socks, so you need to, activate a gene, you can go, right, okay, I want deer socks today. You go take it. But as we keep doing it, things get messed up. Even if we try to put it back the same way, it just doesn't work. So that may be the old epigenome. And the challenge is how we reverse this process. And what we found, so here is a comparative study when we analyzed DNA repair in multiple species. So here, different rodents with different lifespans. We measured the efficiency of DNA repair, and what we found was that in the way DNA was repaired correlated very strongly with maximum lifespan, which means those critters that live longer, they repair their DNA much better. And we wanted to find, okay, how they managed to do this. And we found that one protein called sirtuin-6 or SIRT6 was very important for it. And this protein was responsible for, in a major part, for having more efficient DNA repair in long-lived animals. And then we thought, well, is it also true in humans? What about long-lived humans? Because there are some people that can live more than 100 years. And you're very lucky if one of your relatives is long-lived, because that means you may be inheriting very good genes. So here is a family of centenarians. So here you can see this lady is 115 years old, and there are 5 generations of her descendants, her daughter, granddaughter, great-great-granddaughter, and this is great-great-granddaughter. And that's amazing. We all want to be, as lucky to live very long. And also these people they remain healthy for a very long time. So it's not only, well, she lived 115 years, but she remained active for the most part of this time. So there is this so-called compression of morbidity. Eventually everybody dies, but many of these centenarians, they only become frail and disabled just before death. They don't spend decades in this state. So that's something we want for everybody. And if for those of us who were not lucky enough to inherit such genes, maybe we can do something to, pharmacologically to help everybody achieve this healthy longevity and live as long, and healthy life as centenarians. So we studied genes of the centenarians, and this was in collaboration with my colleague at Columbia University, and we looked at the same genes, sirtuin-6, that we just found in the animals, that it's important for long life and for good maintenance of the genome. And we found that this gene in centenarians has, I mean, not in all centenarians, but in some of them, they are very lucky to have change in this gene. So two amino acids are different. So we were very interested, like, does it make this gene better? Can this explain, why these people live longer? So we took this gene from centenarians and we tested how good it is in DNA repair. And this is an assay. I will not go into details. We don't have time for that. But we tested how good it is at repairing DNA, and we found that it is better than the common gene. So this gene from centenarians, that version of the gene was actually better at repairing DNA. And then there is another very important function that sirtuin-6 has. It can control repetitive elements, so-called line and sign elements. So what are these? So when you think about human genome, you think about it, as well, it has important genes for our function, but it also has a lot of junk in it. And this junk may be some remnants of viruses that integrated into the genome, maybe millions of years ago. So the genes only make this 1%. And more than 30% are these elements. Some of them are parasitic elements. So we want to keep them silent. And this is actually part of the genome that we want to keep quiet. But as we get older, it actually starts to open up and raise its ugly head. So what sirtuin-6 does, besides helping DNA repair, it also helps silence those parasites. And what we found that the version of sirtuin-6 from centenarians was better at doing it. So here we measure how active the parasites are in sort of wild type, like common version of sirtuin-6. We make it like, well, this is our reference. And when we put sirtuin-6 from centenarians, it was keeping it down. So what we show basically is that sirtuin-6 not only helps repair, not only is more active in animals that are longer lived, but also in humans that are longer lived. And it is better at repairing DNA, it's better at keeping those parasites quiet. So then now, of course, the question is, how do we make it more active in those of us who did not inherit this version of the gene? And what I want to show you is that we decided, okay, let's just, as a proof of principle, if we take sirtuin-6 and put an extra copy into human cells and see, will it make the cells younger? So here we took cells, like skin cells, from older people age 60 to up to 90. And we can actually, there is a way to measure biological age using DNA methylation. So DNA methylation is kind of a mark on DNA that is very important for knowing which genes, to activate, which genes to repress. And as we get older, these marks also start to be messed up. And it is possible to design a clock based on those methylation marks. And that was like one of the big discoveries in the past decade, that people designed such clocks. So Steve Horvath was one of the first inventors of the methylation clock. So we could measure biological age of our cells in the beginning of the experiment and then measure it again in the end. And what we saw that when we added one more copy of sirtuin-6, methylation age went down. So we rejuvenated the cells. So that was extremely exciting. And then we thought, okay, how do we make it accessible to people? And of course, this was a genetic experiment, gene therapy. We gave extra copy of the gene. And that is possible So we're actually working on gene therapy approaches for this. But this is, of course, a little bit more dramatic, right? And maybe some risks associated with it. So we would all want perhaps some pharmacological way to also activate SIRT6 and maybe with something very safe. So we started looking at various molecules that can activate SIRT2 and 6, and we tested a variety of such molecules because SIRT2 and 6 is an enzyme, so we can measure enzymatic activity. We give it various molecules and we'll see whether it increases enzymatic activity. And we found one molecule that was making SIRT6 maybe 10 times more active. So it was very interesting that this molecule is called fucoidan. And fucoidan is isolated from seaweed, especially brown seaweed, something like this, like kelp. And it's very interesting that people consume seaweed as food, and seaweed is very popular food item in countries such as Japan and South Korea. And in those countries, people have the longest life expectancy. So we thought, oh, well, maybe seaweed contributes to that. So we decided to do experiment in mice. We purchased old mice to better mimic situation with people, because if we want to have some anti-aging treatment, we're not going to apply it to children. We want to give it to people who are motivated to take it and who need it most, so people in the sort of second half of their lives. So we took mice that were 14 months of age, which would be like maybe human, 60-year-old human, and we gave mice supplement of seaweed in their diet. And then we measured their frailty score every month. So frailty score is very similar to geriatric frailty assessment, where we measure grip strength and walking speed of the mice and condition of their fur and eyes. And what we found that mice that received seaweed supplement, they lived, not lived, but they had lower frailty scores. So, of course, with age, frailty scores were increasing, which is what we expect, but they were increasing less in mice that received seaweed supplement. Then we also looked at the blood of these mice, and here we measured various inflammatory molecules, like interleukins, everything that corresponds to inflammation. And we saw that mice on the supplement had lower inflammation, so which is a very good thing, because inflammation drives various pathologies, from arthritis to neurodegeneration, diabetes. There is inflammation everywhere. And finally, when we looked at the lifespan of these mice, we see that, well, this is a familiar now survival curve. We start with 100%, and then over time, mice die. Mice on the supplement were living longer. So now we are thinking, well, maybe it's time to also try this in human patients. And we know seaweed is very safe. Not every preparation of seaweed has enough hyaluron to activate SIRT2 and 6. So we want to start with the batch that's very good at activating SIRT2 and 6. We can give it to people. We can measure frailty scores. So this is something we plan in the planning. And to wrap it up, what I would like to say is that, well, we have this process of aging and aging of our epigenome. So you see the socks, and that gets messed up with the sock drawer. So now we are looking for ways to reverse the clock, to rejuvenate and put the epigenome back together. And SIRT2 and 6 activation may be one of those strategies. And of course, we are looking at various other ways to turn back the clock, slow down aging process, and improve human lifespan, and most importantly, health span, so that people stay healthy for longer. And this is my wonderful group of people, very talented students, postdoctoral fellows, and we all work together. in finding ways to slow down aging, and also we have multiple collaborators outside of the University of Rochester, and I'm happy to take any questions.
Okay, very good. Let me get my audio back. First, our president's back now. Jim, do you want to try it? Would you like to say a few words? Let me get my video started here. There we go.
Yeah.
Started again there.
Yeah. Hi,
Okay.
everybody. I'm Jim Boyd, president of the Club of Rochester. Thanks for joining us tonight. I had trouble getting in, but as Bob kicked off, this has been a real topic of interest to me really only in the last year, right? I was driving back from South Carolina and surfing around for a good podcast, right? And I found one from Dr. David Sinclair at Harvard Medical School. And I just found it so fascinating. I ended up listening to every one of his podcasts. So for like 6.5 consecutive hours, I listened to, the results of his research on human aging. And so that kind of became a little bit of an obsession for me. And it's like, man, I'd love to find a speaker. Maybe I can find a speaker from MIT, or maybe I can find one, locally. And so I did some Googling and voila, I found Dr. Gorbunova. And so it was great. I listened to the entire thing, and thank you so much.
Let me cut you off for a minute because we got questions. Let me go through them one at a time here, several as a matter of fact. First is, can you define transgenic? You used the term earlier in your talk about mice are transgenic or can be transgenic and people aren't.
Transgenic, it means we put a gene in. So we engineer the gene in these mice.
Okay. So that's all means, nothing more than that.
Well, there are different ways of transgenics, but I guess I don't need to go into these technical details.
Okay, that's fine. Another question from Beck. B.Sc. Well, never mind. The question is: are there ways to sign up for research trials, and are there things people can do now independent of the research trials? I think I can guess what one of them would be.
Well, yes. Right now we are in the process of, getting this trial approved. So it's not yet open for, signing in. But I know that there are various organizations that are now trying to promote more like, sort of citizen-based approach where people can, because you see, there are different types of trials. So there could be a placebo-controlled trial where people sign up and you can be assigned to either placebo arm or the real thing. And then sometimes there is a crossover. So you take placebo for 6 months, people take like a blood test measure, but then there is a crossover. Everybody goes into the trial so that nobody is upset. There are also various kind of longevity clinics where people would take supplements and then just volunteer to report, maybe give a blood sample. So, this is really a rapidly growing field. A very confusing part, and I think that is something we really need to work on, is that sometimes it's difficult to tell apart what's real science, and there is also a lot of snake oil here, because if you just walk into any vitamin shop, you can see like everything is anti-aging, and you don't know if there is really if these claims are supported by evidence or not. Yes.
Go ahead. I was going to say we got several more, so keep going. So let me know when you're finished with your question, answering that.
So no, I just want to sort of a word of caution that there are many unsupported claims, and it's very confusing often, even for people who are in the field. There are certain claims we really don't know. I mean, does it really delay aging or it doesn't? So often, my judgment here is like, first of all, it needs to be completely safe. And with, in good consciousness, I can recommend things like seaweed because I know it's safe. But there are, of course, many other supplements that, there is maybe limited evidence for some. And so people just really need to use their judgment because some things are not 100% safe. So you need to be very careful.
Okay, well, that leads to that moves right into the next question, which is, are any of the commonly available edible seaweeds kombu, kame, high in SIRT6 activation? In other words, what should we go out and buy?
Well, I don't want to turn it into advertisement campaign. So, you could just buy seaweeds and make soup with it. I also test batches of seaweed and for the company called Do Not Age. So it's like DNA, Do Not Age. And what they sell, we test in the lab that it actually activates SIRT6. But, there are also other sources of fucoidan you can buy. And there is about 30% chance they will activate SIRT6 as well. So I don't want to, make advertisement.
Very good. I've got several more questions there. We're a questioning group here, which is good. Next question. I am an MIT undergrad, but a University of Rochester med school grad. Question. Can you do SIRT6 assay on me to guide me on how quickly to spend my retirement fund? That's a really good question.
That's a very good question. 36, we are really not in a way of, basing a biomarker clock on it yet. That mutation that I was talking about in human centenarians, it's very rare. So I think a chance of any one of us having it is pretty low. Maybe if we have a grandma who lived to 110, the chance would be slightly higher, but still rare. But there are biological clocks. So I mentioned methylation clock, right, which we use to measure what effect of the treatment. And of course, insurance companies and everybody would like to use it to predict, what's life expectancy of people. I can say that although we can use it to kind of guide our, maybe clinical trial on a large group of people, so it generally can measure biological age, but for, let's say, personal decisions of when to take retirement and, how much to contribute into your IRA, it's still not accurate enough. And we would like really to improve accuracy of those biomarkers, but we are not there yet to make individual recommendations because it's just not accurate for that.
Okay. Next question. Okay. Are there any supplements you feel there is good support that can extend your lifespan? For example, resveratrol, NAD, and Elysium.
Oh, yes. Well, there was a lot of controversy around resveratrol. It was, first discovered by David Sinclair, who is my good friend. But then there were also some people disproving the effect of resveratrol. And when it was tested in mice, it didn't really have effect on lifespan. It did help mice that were put on a very unhealthy, high-fat diet, and then resveratrol had some positive effect. But when mice were given regular diet, it had no benefit. So this is why, I don't particularly recommend resveratrol. I don't think there is enough evidence for it. Okay. NAD, yes, it's very popular. Elysium, I'm on the board of Elysium because, they asked me to be on the board. And Lenny Guarente, who is at MIT, is the founder of it. So what can I say? There is some evidence of NAD being supportive of muscle health. I think that evidence is fairly strong. For the lifespan, we really don't have good evidence.
Okay, the next question. There's quite a few of them here, actually. I understand that centenarians have different CD-4 cells. Is this correct?
Well, immunology is a very big and complicated field. And yes, so better immune health is definitely a link to longer lifespan. You need to have good immune system to live longer, but you also need to not be overly zealous immune system, because then you end up with inflammation and autoimmune disease. But we still don't really understand all the intricacies, how it's best to tweak our immune system for optimal longevity. And this is where our bat research comes in, because bats, they have it. They have immune system that is so good for resisting pathogens, and they control inflammation very nicely. So I hope we'll learn from bats.
Okay, a lot to learn from other animals. Okay, next question. Could you clarify the association between Fucoidan and HA? Fucoidan? Yeah, I hope we got it right.
Oh, well, these are the beginning of my talk and the last. They are both polysaccharides, but of different structure. And right now, we don't see clear association in terms of the mechanisms in the cell, because fucoidan is a molecule from seaweed that happens to activate sirtuin 6. Hyaluronan is a part of our tissues. It's, fills space between cells. They happen both to be of a similar class of compounds. Maybe that means something, but we really don't know what the link is.
Okay, I think you answered the rest of it, which is HA a form found in brown seaweed?
No, HA is found in our tissues. It's found in connective tissues, like in your under the skin. That actually what gives elasticity to the skin. We have a lot of HA in the blood vessels. It gives elasticity to blood vessels. In synovial fluid in our joints, HA what lubricates.
Okay, got it. It says also, I believe, this is a quote from the question, I believe you said that fucoidan increases SIRT6. Is that correct?
Yes.
Okay. Next question. That's we've got a lot of good questions here. There's still several more. Can you comment on resveratrol? Boy, that's why I'm not a doctor. I'm an engineer. Can you comment on resveratrol?
Resveratrol.
Well, the way it's spelled here, and it's got a question mark on it next to it, whether it's spelled correctly. Reseveratrol. Resveratrol?
Resveratrol.
Okay.
Comment. Well, I think I commented already.
Yeah, it sounded familiar. Okay, let's move on here. Okay. Has anything in your research touched on factors that would protect against dementia?
Yes, there is evidence that 36 is, and I should mention actually the sirtuin, since we talked about resveratrol and Elysium. So Lenny Guarente at MIT, he was the first scientist who discovered the effect of all these sirtuins on longevity. So I really need to give him credit for that. And David Sinclair came out of his lab. He was his student. And so
sorry for interrupting. But what are your thoughts on David Sinclair's research, recommendations of lifestyle changes, and supplementation, which was the very next question now that you mentioned David Sinclair? So I apologize for interrupting, but it's too attractive.
Well, he's a good friend of mine, and we collaborated. So this idea of epigenome kind of getting disrupted with age. So we both, developed this hypothesis as being the driver of aging. But that's scientifically. Now to his lifestyle advice, maybe I'm not that familiar. I think he probably says things that make sense.
Okay, sounds good. Okay, how many types of SIRT6 are there? Is the specific SIRT6 found in 100-year-olds key, or will increasing any of the SIRT6s be helpful?
Well, so there are different sirtuins. So there is a family of them, like 1, 2, 3, 4, 5, 6, 7.
Okay.
And the one that we studied was sirtuin-6. David Sinclair, actually, in his lab, he did a lot of groundbreaking research on sirtuin-1. But then it turned out that sirtuin-6 is actually more interesting for aging because we have more effects on aging. So now we both study sirtuin-6. It's a protein. That version that we found in people who make it to 100, it's very similar, just different by 2 amino acids. And it gives it a little bit more activity.
Okay, I see. Next question, I think we've already answered. It's what supplements are you? No, this is a little bit different. What supplements are you taking, and would you would you recommend to prolong longevity? I think the second part you answered.
Well,
First part you might not want to answer. Yeah.
I don't really take any supplements at the moment, but I'm trying to eat. Well, I do eat seaweed. This I'm taking. But I'm trying to eat healthy diet, and there are so many different recommendations about the diet, but many of them are not evidence-based. So what we know from research that a diet that is rich in fruits and vegetables, is very beneficial, and especially vegetables such as cabbage and similar cruciferous vegetables, and various berries like blueberries, raspberries. They have molecules in them that have a beneficial effect on lifespan. So you cannot go wrong with eating blueberries.
That's good. Very good. Okay. What do you think of the recent paper in Cells by David Sinclair? It seems to be a popular guy. Let me try that again. What do you think of the recent paper in Cells by David Sinclair's group showing the epigenetic changes may be significantly more important than the double-strand breaks?
Well, I think his paper really, and I happen to know this paper very well, that double-strand breaks actually result in epigenetic change. Because if you have a lot of double-strand breaks, it accelerates the drift. So remember the boat, the drifting boat. And so it accelerates epigenetic drift. So David Sinclair in that paper actually linked those two things. And we've been studying double-strand breaks for a very long time, and we find them to be very tightly linked to the process of aging. And this paper of David Sinclair, really brought these two concepts together. So I'm completely on board with that idea. I know that there were some people criticizing it. In science, there is always a lively debate. But I actually agree with David Sinclair's concept completely. And I can, share with you that I was asked to kind of judge who's right with this criticism, and I actually expressed this to the journal that I think, the concept that proposed in this paper, I agree with it, and I think it's scientifically sound.
The next question, does NMN do the same thing as seaweed?
No, it does different things. So NMN is also a popular supplement. It's part of NAD boosting, chemical. It becomes converted to NAD. And NAD can activate sirtuins. It needs them as a cofactor. But with respect to sirtuin-6, although it needs NAD, you cannot really activate it too much with NAD. It just takes what it needs, and you cannot boost it very much. Mm-hmm. So I would say for SIRT6, fucoidan is a better activator.
Okay. The next question. Would any results from DNA testing, for example, Ancestry 2.3 or Ancestry or 23andMe, be indicative of how long you're going to live?
Well, not really, I would say, because DNA testing, there are different types of DNA testing, because we just discussed methylation age test. And it can give you some prediction. It's still not super accurate, so I wouldn't plan my retirement based on this prediction. But it's entertaining, and if you have a chance to get your methylation age measured, why not? On a large scale, when looking at the population of people, that is actually fairly accurate. But again, as I said before, it may not be accurate enough for individual life decisions. But it looks at methylation, which is like a little mark on the DNA. So you can take DNA and measure those marks. Now, what 23andMe does, they just sequence DNA. They just determine DNA without looking at those marks. And that is good in, telling you about some genetic predispositions, for example, how likely a person could be to develop, diabetes or some other condition, or it's also fun for ancestry. But it cannot really tell you how long you live, because, it's interesting. That could be a topic of another long discussion, that our genes determine about 25% of how long, we'll live. And then there is a big contribution from the environment. And now, I mentioned several times during today's talk about centenarian. So if you have a grandma who lived to 100, so then you might also live to 100. Because for those people who have this exceptional longevity, the heritability is much higher. It may be more than 50%. But for sort of an average person, it's maybe around 30%. So that's why just by looking at DNA, unless it's a DNA from a centenarian, it's not very predictive of your longevity.
Okay, very good. Next question is, are people able to have their methodological, hope I got that right, clocks assessed? Is it something that would be affordable or covered by health insurance?
Well, yes, you can get it assessed. It is not yet covered by health insurance, and I hope it will be at some point. We'll probably need to make it a bit more accurate for insurance to start covering it. And we will need to also perhaps show that it doesn't just tell your biological age, but maybe it can predict some, disease onset, and this way people could take certain steps to prevent it. So because, insurance usually doesn't cover something just for curiosity. They would want to say, okay, based on this, you can do this and that. So right now, we're actually working on a project to develop a more affordable methylation age testing and then link it to health outcomes. And this way, maybe every time people walk into doctor's office, they would, do blood work. They also do this methylation test. And you can even, let's say, if you started some health routine, it may tell you, well, are you on the right track? Is your biological age going down or going up? So I'm actively involved in this research, but you can find some methylation tests online. And Steve Horvath, who was one of the, first discoverers of this method, he has, I think this is called the Clock Foundation. You can probably order a test from them. The cost is around $200, maybe $250, which, it's not cheap, but, if you're curious, you can do it.
Okay, the next question is for me. It says, will you be sending a link of the recording to the attendees? And the answer is absolutely. This is being recorded, and given that we had our technical problems at the beginning, I want to be sure that people that really were interested in hearing what you had to say at least get to hear it afterwards. So I will be sending the link to everybody who registered. You will get it. Okay, the next question. What do you think is the best diet for life extension? David Sinclair supports a vegan diet. Vitamin B12 supplementation is an optimal diet. Do you agree?
Well, vegan diet, there is a link between consumption of red meat, or maybe more specifically processed meat, and shorter life expectancy. Although in young people, I would not recommend vegan diet. It is, there are health detriments. Vitamin B supplementation may help partly, But I think for young people, especially, growing their muscle mass, vegan diet is very bad. If you are like my age, then that may not be a bad idea, limiting your meat. And often when you get to that point, you really don't crave meat as much as before.
I'll say, how about my age? Okay, good.
Yeah, so I think that is reasonable to limit consumption of red meat, past age 50 or 60. But I would not really advocate for strict vegan diet.
Next question, let's ask a follow-on on your answer about dementia. It says, has your research touched on factors that would protect against dementia?
Oh, that's right. I didn't finish that. We switched to David Sinclair. So that's evident that sirtuin-6 level is lower in people with Alzheimer's disease. So I hope that if we develop some strategies to boost sirtuin-6, it may help dementia also.
Oh, okay, that's good. Okay, is the relationship between red wine and resveratrol? There it goes again. Can you comment?
Yes, there is a relationship. So resveratrol is found in red wine. But, the sad part is you need to consume too much red wine to have a meaningful amount of resveratrol. And on top of that, I don't think the evidence for resveratrol is strong enough. So I think, you can enjoy a glass of wine. And there are many other molecules in it that may be helpful. But again, there is still a bit of a debate because, there are some compounds in red wine that may be beneficial, but having too much alcohol also has detriment. So, I think here there are different studies claiming different things. Some people would show that moderate consumption, or some studies show that moderate consumption of red wine actually is associated with longer life, but then others say like any alcohol is detrimental. I think the jury is still out on this one. Okay.
Next question is, we're getting near the end now, so you've been very patient, very good. Thank you very much. Do you teach a class, undergraduate or graduate on this subject, or is it only research? Is it research only at this time?
No, I teach a graduate and undergraduate class at the University of Rochester. Yes, it's called Biology of Aging.
Were there any classes at MIT? I bet there are. There should be, given the group over there. Okay. How important do you consider telomerary, TELOMERE depletion?
Yes, well, that's an interesting question. So telomeres, so these are the ends of chromosome, because our DNA is arranged in like linear chunks. But at the very end, there are loose ends. Right. And as we get older, they shorten a little bit. And there was a lot of excitement about telomeres. There was a Nobel Prize awarded for telomere research several years ago. And what was discovered that this shortening of telomeres, well, it happens over time. And what's interesting that in cancer cells, because, it's not that they shorten to make us mortal. I mean, initially people thought about, but this is not the point. They can shorten to protect us from cancer, because if we get a pre-malignant cell, it starts dividing, right? But then it shortens its telomeres very quickly, and then it runs out of it, and it actually stops, because that makes it impossible for this cell to grow any further. So telomeres shorten to protect us from cancer. But at the same time, right, if they shorten in other tissues, that may be bad, because that also may contribute to aging. And there is a very weak correlation between telomere length and age. This correlation is even weaker than, we talked about biological clock, that DNA methylation, that is not accurate enough for retirement planning. So I would say telomere length is even like orders of magnitude less accurate for retirement planning. So there is some correlation, yes, but it is very weak. And I would say, it does contribute to aging, but this is really not now the prevailing sort of way of looking at aging, and even like trying to make them longer. It's not going to solve aging.
I see. Okay, the last question that's been written in is, what is the role of autophagy, A-U-T-O-P-H-O-V-Y, autophagy, and longevity? Does CWIT affect autophagy?
Yes, no, it's an interesting question. So autophagy. So this is the process of the cell cleaning up itself, because when like within cell there is some waste, like there could be some component of the cells that go bad, and they need to be utilized, and then there is this self-cleaning system called autophagy. So it is very important. It is very important for longevity, and if it goes wrong, then the cell becomes filled with junk. It cannot clean itself up. And this is more important for cleaning up proteins, because my lecture today was more focused on the DNA and like epigenome, but you also need to keep up your proteins. With proteins, because they can be replaced, so this autophagy, it would take proteins and recycle them and make new ones. So in a sense, you can kind of rejuvenate your proteins many times over, but with DNA, it's much more difficult. There is only one DNA strand, like you cannot just redo it easily. But still, like if this recycling system starts failing, cell is filled with junk, and that's what happens in many forms of dementia, because neurons are cells that live for a very long time, and then they start to accumulate junk, and that creates, Alzheimer's disease, those plaques and tangles in the brain. So it is a very important process. Whether seaweed affects it, we never tested, but that may be something interesting to us to test.